Good evening, madam.
Good evening, sir. Sir, shall we start the class?
Yes, ma'am. Yes.
Vinay, sir.
Madam. Cheddam, madam.
Start cheddam, sir. Live start.
Adhe nenu andukane start cheyadam.
Okay, sir. Start cheddam.
Sir, good evening.
Acha. So, good evening to dear participants. So, today's topic is on micropropagation, that is tissue culture techniques in banana. So, Dr. Ravindra Kumar, he is the expert in tissue culture. He is having a lot of experience in tissue culture. So, he has completed his BSc from College of Horticulture, Rajendra Nagar, Hyderabad, and he has completed his PG and PhD from IARI in the Division of Floriculture and Landscaping with ICR, JRF, and also SRF fellowships. And he has started his career as a horticulture officer in Department of Horticulture during 2005, and later on he joined as scientist in Dr.YSRHU during 2009. As a scientist, he worked at HRS Chintapalli, Ambajipeta, and also Kovvur on black pepper, ginger, turmeric, coconut, cocoa, banana crop improvement, and also production aspects. And he has been instrumental in establishing the new commercial scale banana tissue culture laboratory at HRS Kovvur. As a tissue culture specialist, he has standardized various tissue culture protocols for commercial sale and multiplication, crop improvement in different crops like banana, pineapple, pomegranate, coccinia, colocasia, chrysanthemum, marigold, zerbera, tuberose. Apart from Grand Nine, he has standardized tissue culture protocols of native banana genotypes of AP by using apical meristem and also immature male flower buds as explants. Till now, he has guided 11 PG students and 6 PhD students as chairman and also advisory on as an advisory member on micro propagation aspects in different horticulture crops. He is associated in release of 4 coconut varieties and also 1 greater yam variety. He has published 56 research articles in national and also international peer-reviewed journals. He has also participated in 30 national and also international conferences and also presented 92 research abstracts. And he is a life member for 3 scientific societies. And he is also the recipient for ICR IARA Merit Medal, ICR Jawaharlal Nehru Award, ISOH Best Thesis Award, and 5 Best Oral Presentation Awards in different national and also international conferences. At present, he is working as senior scientist at HRS Kovvur and working on standardization of low-cost banana and also tubers and also economically important horticulture crops in micropropagation protocols and also for the crop improvement of the different horticulture crops. So with this brief introduction, I request Dr. Ravindra Kumar sir to present the today's topic on micropropagation techniques in banana.
Thank you very much, Dr. Vinay, for your good words. And I will share my PowerPoint. Hope my PowerPoint is visible.
Yeah, yes, sir, it is visible.
Okay. Shall I start?
Yeah, yes, sir. You can start.
Good evening to the dignitaries present for this Banana Graduate Readiness Program lecture series. And very good evening to all the enthusiastic students. Today, I'm going to deliver my topic on micropropagation techniques in banana. And before going into banana and micropropagation techniques, just to see the history of plant tissue culture, there are so many things happened from 1902 onwards till date. But the first initiation taken by Haberland, he has proposed the concept of in vitro cell culture during the year 1902. Later on, so many hormones and other things identified. Again, 1957, Spoog and Miller identified kinetin and the cytokinin mode of action. So this is a major achievement. Again, for commercial plant tissue culture, during the year 1962, Murshige and Spoog developed the MS medium with a higher salt concentration, and again they have modified this MS media by increasing the sucrose concentration. So these three are major achievements for today's commercial micropropagation of different horticulture crops or any other crops. Coming to banana in vitro culture, during the year 1960, Cox et al. They have successfully cultured the zygotic embryos of some banana cultivars in vitro means. And during the year 1964, Ram et al. They have started a parthenocarpic green fruit pulp of several clones in vitro. They have obtained a calyx but failed to regenerate the shoots. The first in vitro commercial multiplication protocol of banana was reported by Ma and Shi in Taiwan during the year 1972. Later, Berg, they have standardized the elimination of cucumber mosaic virus in banana by using meristematic tip culture and heat therapy combination. Guzman and et al. They have started irradiated banana shoots and culture. And due to these efforts, during the year 1990-91, total 15 to 20 million tissue culture banana plants raised worldwide for commercial multiplication. In 90 itself, the banana commercial multiplication was started in the globe, and there onwards, the journey is continuing, and so many labs established worldwide and involved in production of tissue culture banana plants. Coming to Indian status, banana tissue culture in India, the first commercial micropropagation laboratory in India was set up in 1987 at Cochin Export Processing Zone under A.V. Thomas and Co. Company. And it was followed by Indo-American Hybrid Seeds at Bangalore, Hindustan Labour Limited at Mumbai, and Unicorn Biotech at Hyderabad during the year 1988. During the year 94 and 95, Jain tissue culture laboratories were established at Jalgaon, the biggest laboratories, and have production capacity of 85 million plants per annum. At present, they are multiplying Grand 9, Nandran, Elakki, and different banana cultivars also. Followed by this biggest laboratory, one more large scale, more than 10 million plant capacity laboratories are only two. That is, one is Jain Laboratories, and second one is Regen Shine Biotech. It's existed in Pune, Maharashtra. They are producing nearly 30 million banana tissue culture plants. So, if you see quality planting material, it's fortunate that banana having a tissue culture efficient tissue culture production system. So, in India, majorly bananas are cultivated by tissue culture. The two Cavendish genotypes are mainly cultivated by using this procedure. But if you see the installed production capacity of our tissue culture labs in India, they can produce 650 million plants per annum. But some hurdles, some difficulties, and some other reasons, the production capacity is actual supply is only 455 million plants per annum. In India, banana cultivation reached to 9 lakh hectares, and the largest banana producer and consumer also. So, the requirement of quality planting material in the form of suckers or plant tissue culture plants required 2,500 to 3,000 million plants are required. So the gap is huge, more than the 6 times. If we increase the production capacity, then only we can achieve the target. So lot of scope is there in banana tissue culture industry establishment for young graduates like you. So you can learn the techniques and implement. You can implement in future for your development and creating employment in the form of establishing a industry. So if you see, according to NCS TCP, that is a national certification system developed by DBT, only 60 to 70 laboratories are accredited in India. Still 100 more laboratories are working, but they are not accredited. So from 0 to about 10 million plant material production capacity, we have so many labs are there. About 10 million, only 2 laboratories are there, that is existed in Maharashtra. In our Andhra Pradesh, Hyderabad is main hub for this physical plant material production. And recently in Anantapur, Nandyala, other areas, Kadapa, some enthusiastic tissue culture persons came and they have established the laboratories, commercial scale laboratories for quality plant material production. Before going for tissue culture techniques, you know what is tissue culture? The collection of experimental methods and for isolation and inoculation of organs, tissues, and cells in an artificial medium under in vitro aseptic conditions. So it's like a vegetative propagation only by using a in vitro means. So vegetative propagation in laboratory, you can tell like that. In general, bananas are generally propagated by sword suckers in commercial fields. But some of the varieties like Assam wild and other genotypes we can multiply by using seeds, as they can set seeds and we can easily multiply it by using seeds. Otherwise, all the genotypes of banana, most of the genotypes belongs to triplets, so they are sterile. We can multiply by using only vegetative means, that too sword suckers. So before as we know that sword suckers, through sword suckers we can multiply these planting material. We can multiply the banana, but why we need tissue culture? We have to think. If you multiply by using suckers, the limitations are so many. The first one is the multiplication rates are very low. The the plants are non-uniform in nature. Productivity is very low. And the spread of various banana pest and diseases are more. If you see in banana, 4 major virus infections are there. That is the banana black mosaic virus, cucumber mosaic virus, and banana bunch top virus. So these viruses can spread through planting material, through suckers. And some of the deadly diseases like banana bacterial rhizome rot caused by Erwinia and Panama disease caused by Fusarium, they can also transfer by through this planting material. And the nematode infestation also, nematodes can also nowadays is a major threat for banana and other horticulture crops. They can also transfer by through this planting material. So if you use suckers, infested suckers as a planting material, the productivity will be reduced and lands will be contaminated. So those lands contaminated with this bacterial rhizome rot and Panama wilt, not fit for banana cultivation in future. So in vitro clonal propagation, that is tissue culture plants are recommended for, highly recommended for planting. Similarly, if you see the age of the sucker, to use a matured sucker, it will give crop 8 months or 9 months. To select peepers, that is young suckers, it will give bunches at 14 to 15 months age. So the farmer selected different kinds of suckers. You should wait from 8 months to 14 months for continuous staggered harvesting. That is highly problematic for export or other things. For cultivating other crops also not possible with using this sucker. And if you see some of the advantages of tissue culture planting material, they are true to type nature. Whatever the plant material generated by using this technique, they are similar to mother plants. So if we select a good mother plant, all the plantations generated from mother are very good and true to type in nature under well management conditions. And these are quality planting material, relatively free from any kind of disease or pest. As we will do virus indexing and we will multiply in laboratory conditions and we will do hardening in a restricted environment. So these are completely free from any sort of pest and diseases. And as all the regenerants are having same age, they will grow equally. So uniform growth and higher productivity is possible by using this tissue culture plants. Early maturity of crop and maximum land use possible in low land holdings like our country. That means precocious bearing. Early bearing we can expect by using this tissue culture plants. Unlike suckers, we can multiply these tissue culture plants all year round and available throughout the year. We can supply the quality planting. Due to this precocious bearing in nature, we can get 3 crops, 1 plant crop and 2 rotten crops in 2 years period, like in cultivars like a grandname. Otherwise, it will take 3 to 4 years for completing 3 banana crops. We can save the resources as all the plants will come to harvesting within a month. And unlike the suckers, tissue culture plants, 95 to 98% of the tissue culture plants bear good quality bunches. Earlier in sucker propagation, only 75 to 80% of the plants bear bunches. So farmer can enhance their income by increasing the productivity and higher quality bunches. And the new varieties can be introduced and multiplied in a short duration. Suppose now gramine is highly susceptible for this Fusarium tropical race 4. So if some other better gramine variety resistant or tolerant for this race 4, tropical race 4, you can multiply these new varieties immediately by using this tissue culture technique. Whereas through suckers, it is highly impossible. Coming to techniques of tissue culture, before initiating banana tissue culture, the selection of superior mother clone is a highly important aspect. Because if we not select, fail to identify the true mother plant, whatever we multiply, the characters will represent in field. So if you select good mother plant, all your plantations raised by using those tissue culture explants are uniform and will give good results. If you select poor mother plant, all the plants will give poor growth and development of it. So care must, you should take good care to select a healthy and good mother plant which is relatively free from any sort of pest and disease. The mother plant should represent the particular banana group which it belongs in. Banana, so many groups are there, so the characters should be similar. And as per our experience, we recommend to collect explants only from plant crop. Plant crop suckers are relatively having free from any sort of pest and diseases. In ratoon crop, the quality may decrease. In banana, if you see, there are so many groups are there, like Cavendish, Kovvur, Rastali, other groups are there. Out of 100% total cultivation, nearly 44 to 50% of area is cultivated. This Cavendish group occupied the total banana area. Half of the area occupied by this Cavendish group, followed by Kovvur group. While selecting the mother plant, we have to check that the characters of that group should be represented. Grandine means the finger length, finger girth, yield, height of the plant, blotches on the plant, all these things you have to look before selecting the plant. After selecting the mother plant, our main objective is we have to produce disease-free plants. As we already know that 4 threatened viral diseases are a major threat for this banana cultivation, we have to do virus indexing to eliminate these deadly viruses from the chain of plant material production. So we have to use double antibody sandwich, that is DOS ELISA method for detection of cucumber mosaic virus and banana bract mosaic virus, and two other diseases like banana bunchy top virus and streak virus can be identified by using PCR based techniques. So in our area, mainly this CMV and bract mosaic virus are highly dangerous. So if you use antigen and antibody technique, if they react and give the spectrophotometer reading more, that means the color of the buffer will change. So if the color of the buffer changes, the samples notes that it is infested with banana bract mosaic virus or CMV mosaic virus based on the specific reaction. So we have to collect the suckers from the mother plant. Those buffer reactions, body values came 0. So we have to collect those plants, those suckers only, and those suckers will be relatively free from any viral contamination. Immediately we can culture, or we can keep it under our greenhouse or shaded conditions for further multiplication in invert. The first step after collecting the explants are inoculation, initiation of shoots. So relatively you can tell commonly in normalized way, you can tell that we can take any plant part of banana for commercial multiplication, but our research study is proven that plant crop suckers of 1 to 1.5 kgs stored suckers are highly suitable for selection of explants, and the height of the sucker might be 40 to 100 centimeters, be free from pest and diseases. Weight should be more than 1 kg. And the representative of all the characters of the relative group. Then we have to remove the pseudo stem layers, and we have to reduce the size of the rhizome to 2.5 centimeter cube. It should contain apical meristem in its portion. Then the size of the explant we have to reduce. We have to take as a cube. You want more shoot multiplication, more auxiliary buds are needed. So if you select a large sized buds, the rate of multiplication is rapid. But at the same time, the chance of microbial contamination and the chance of high phenol oxidation and the blackening of culture is very high. So 10 to 15 mm size explants we generally will select, and if we want to, our objective is to eliminate the viral infection in banana, then we have to select the meristem tip culture as the preferred option. But most of the times, the reduction of meristematic size will lead to mortality, higher mortality rate. If you see the pictorial representation, after selection of virus indexed suckers, then we have to remove all the outer layer. We have to part the rhizome. Then we should bring into the optimum size of 2.5 centimeter cube. Immediately, we have to dip these explant into citric acid solution so that it will prevent the blackening of suckers, and it will prevent the oxidation, polyphenolic phenol oxidation, and prevent the blackening of explants. Then we should wash under running tap water up to 20 minutes, bring it in the laboratory conditions, remove one more layer of pseudohyphum leaf whorls, and reduce the rhizome portion and transfer into the carbendazim 2 grams per liter plus 8 hydroxyphenolane 200 ppm plus Queen 20 solution for at least 15 to 20 minutes. Generally, we use one antibiotic like a plantamycin and streptocycline here, but due to recent ban on antibiotics, I'm not recommending any antibiotic. Generally, instead of antibiotic, I'm recommending 8 hydroxyphenolane for reduction of bacterial contamination. After selecting the 2.5 centimeter cube of explant, before going to laminar bench, we have to remove the 2 outer layers. In the laminar airflow, remaining 2 outer layers we have to remove to inoculate the suitable size of explant in banana. Coming to the pre-treatment and surface sterilization of explants, After this fungicide and bactericide treatment, we have to bring the cultures under laminar airflow and wash thoroughly with sterilized RO water 3 to 4 times. Then remove the one layer of outer layer and disinfect with 70% ethanol for 2 minutes, followed by mercury chloride 0.1% mercury chloride 18 minutes first time. Then remove one layer, treat with antioxidants like citric acid and ascorbic acid. Then again treat with mercury chloride 0.1% with 12 minutes and remove one more layer, and then explants treat with 0.1% antioxidant like citric acid, disaccharidamide. Then inoculate these cultures. You see under laminar airflow, gradually you should remove the outer layers and reduce the size and bring the explants into optimal size. Then treat with antioxidant solution, wash thoroughly, and dry these explants by using the sterilized tissue paper. Then culture into suitable media. Now we have disinfect the explants. But if you see literature or any banana books, protocols are highly different. It's highly depending on the laboratory conditions, the experience of the people, and the land pattern in the adjoining areas, and the cultivars. So many factors are depending on this sterilization and pre-treatment protocols. We have developed our own protocol. In our protocol, what I mentioned earlier, we can able to achieve almost 72% culture establishment, which are free from any bacterial and fungal contaminations. More than 90% culture establishment we obtained in the initial I0 to I1, I2 cycles. And like this, from sucker to culture initiation, if we complete this step with perfect measures, then the rest of the things are easy. As once we disinfect the explants properly, then we will free from any sort of microbial, bacterial, fungal microbial contamination. Then the shoot multiplication will start. So we have disinfected the explants. Now we should disinfect the medium, that is sterilization of medium. Earlier we discussed that Murshik and Spook media generally used for this banana tissue culture. It is composed of macronutrients, micronutrients, vitamins, amino acids, growth regulators, and sucrose. Generally, sucrose is used as 30 to 40 grams. And hormones like cytokinins and auxins will use. Cytokinins generally increase the shoot division, cell division, and enhance the shoot multiplication. Auxins generally used for rooting and other things. In the initial steps of banana tissue culture, auxin requirement is very less. The rooting stage, the cytokinin concentration may be lowered for best rooting. So the balance between the cytokinin and auxin concentration yields the good multiplication and good rooting conditions in a banana. Semi-solid media we have to prepare for this banana tissue culture. So for preparing the semi-solid media, agar is used as a gelling agent, 5 to 8 grams. And coming to the calculations and composition of banana media, generally we used Murshik and Spook media, and the recommendations he has already published. That is a macro element, minor micro element, iron, vitamins, sucrose, and other things he has mentioned. For suppose, as per Murshik and Spook media, for preparation of 1 liter culture media, we have to add 1650 milligrams of ammonium nitrate, 1900 milligrams of potassium nitrate, and so on. But every time, if we weigh and add these elements for each liter of culture media, it's time consuming and it's very difficult also. So we have to prepare the stock solutions. For suppose this macro salts, 20x concentration if we prepare, 1650 into 20 times we have to weigh. So 33 grams of this ammonium nitrate we have to prepare stock. For each chemical If we prepare stock solution, again the requirement of glassware and the storage conditions, very large storage conditions are required. So we can mix all these macro elements as a one stock. So we can mix ammonium nitrate, potassium nitrate, calcium chloride, magnesium sulfate, potassium dihydrogen orthophosphate, all these things we can mix in one stock solution with 20x concentration. So for preparing 1 liter culture media, how we should take how much quantity we should take from the stock solution? That is a question. So as we enhanced 20 times the concentration, we have to take we have to divide the 1 liter media, that is 1000 ml we want to prepare. So divided by the stock concentration, 20 times we have enhanced. For example, if we want to add ammonium nitrate, the macro salt, major salt concentration, how much stock we have to take? We have to divide 1000 ml of stock divided by 20. As 20x concentration we have multiplied. So 50 ml of stock solution we have to take from this major salts. So in this 50 ml solution, we have 1650 milligrams of ammonium nitrate, 1900 milligrams of potassium nitrate, so on, 450 milligrams of calcium chloride, so on. In case of micro elements, the quantity is very low. So that's why you have multiplied with 200x. Instead of 20x, we have multiplied with 200x. So 1000 divided by 200 is 5 ml. If we add 5 ml of stock solution, all these potassium iodide, boric acid, manganese sulfate, all these things will be available. Like this, we have to calculate the major minor stocks, vitamins, and other stock solutions. Generally, in research purposes, we use double distilled water as a water source. But commercial tissue culture laboratories, we use reverse osmosis water, RO water, cheap and can be available more in quantity. So we have to take 700 to 750 ml of RO water, then add all these stock solution concentrations, including growth regulators, BAP, and indolystic acid, like growth regulators, so you have to add, then add sucrose, then make the volume up to 1000 ml. Keep this media on stirrer, magnetic stirrer, and take the reading of pH meter. If you prepare a good media, generally with these combinations, the pH will denote 4.5 to 5. Then our required pH is 5.8. In that 5.8 pH, the banana cultures will multiply very good. Banana and other horticulture crops will perform very good at 5.8. As at that pH, all the minerals available in the media are available for explants. So we have to adjust this pH by using one normal sodium hydroxide solution. For suppose the pH levels are high, we have to decrease the pH level by using one normal hydrochloric acid. Then we have to boil these media on a gas stove. After a little bit warm condensation of this media, add agar the required amount 5 to 8 grams per liter, add based on your standardization. Then we have to autoclave this culture media at 121 degrees centigrade by using 15 psi pressure for 15 to 22 minutes. That is based on depending on the quantity. If you sterilize more quantity, it required more time for sterilization. If you sterilize less quantity, the less time is sufficient. The autoclave is worked based on our pressure cooker principle. Then we have disinfested suckers as an explant, and we have autoclaved media, but the suitable environment we required, bacterial and fungal free environment we required. So for that one, laminar airflows are highly required. In this laminar airflow, initially beneath the portion that in front of our legs, pre-filters will be available. Before beneath that, two motors will work. The motors will suck the fresh air through pre-filter and push from the above HEPA filter. These HEPA filters are having 0.22 micrometer pores. Through that pores, none of the microbial organisms like fungal spores, bacterial spores, hyphae, all these can be prevented. So the laminar airflow environment is completely sterile and free from any microbial contamination. In that sterile environment, you should transfer this disinfected explant into sterilized media. So after inoculation, we have to maintain these cultures in incubation room in optimal light and temperature conditions. Generally, tissue culture labs, particularly banana tissue culture labs, the required temperature is 26 plus or minus 2 degrees centigrade with 12 to 16 hours photoperiod light, remaining dark period, and 3000 to 4000 lux lighting is required. If we provide these conditions in these environmental, then the explant development we can see. And air conditioners are required to maintain the temperatures in our tropical environment. Polar parts incubators generally used, heating incubators generally used for this maintenance of laboratory. But the things are not as I mentioned. There are so many difficulties most of the laboratories are facing, especially the exudation of phenols, and during the light, in the presence of light, the phenolic compounds are turning into oxidation process, polyphenolic light reaction, photooxidation, it's converted into polyphenolic components. These polyphenolic components are black in color and surrounded and form a layer around the explant and prevent the nutrient absorption from the media. So eventually the explants will die. To prevent this oxidation and phenol exudation, the initial stages of banana tissue culture, we have to frequently change the media in 1 or 2 weeks. And one more method is adding antioxidants like ascorbic acid, citric acid in the culture media, and the concentration of 10 to 150 milligram per liter will also help. You see, due to this phenol exudation, the explant, the media is completely changed to dark in color, and the explant also almost in a death condition. And to prevent that one, so many methods are there. Supplementing the media with the charcoal and frequent transfer of explant in the culture conditions, adding polyvinyl pyrrolidine, all these are techniques are available. But at our laboratory, we have extensively studied for this phenol exudation in different native banana cultivars, and we found that addition of 100 milligram per liter antioxidants in the form of 50 milligrams ascorbic acid and 50 milligrams of citric acid is highly effective, and we can successfully eliminate the phenol exudation, and supplementing this phenol exudation will result in a higher culture establishment. You see, in the control, the media is completely black and the explants are going to die. So, if we supplement the media with 50 milligrams of ascorbic acid and 50 milligrams of citric acid, it will prevent the oxidation and keep the explants alive in gramine and most of the native cultivars of banana. One more technique, we found that type of explants also here important. Generally, we have tested 8 different types of explants like a plant crop, peepers, plant crop, matured suckers, ratoon crop, and directly used, first plant in greenhouse or shade house, then use. Among all these treatments, we found that pre-germinated suckers of plant crop of 1 to 1.5 kg weight are highly responsive, and they are giving a very good response. Their survival is more than 98%, and immediately within 1 week, you will observe the shoots sprouting these type of explants. So, pre-germinated suckers of plant crop are highly effective in banana plant tissue culture. And coming to other problems like insoluble precipitate, it occurs mainly due to the salt formation between the elements what we use, like calcium, magnesium, phosphate. All these reactions will happen, and insoluble precipitate will form. So to prevent this, first we have to, while preparing the media, First, we have to add nitrogen compounds like potassium nitrate, ammonium nitrate, like that. Then we add magnesium sulfate, then calcium chloride. Ultimately, we should add dihydrogen orthophosphate, phosphate form. So, sequentially, if we add these elements and calcium as a separate stock, will prevent the insoluble precipitate. And one more thing is non-availability of ammonium nitrate. In the next coming slides, we will discuss how to overcome this problem. And other problems like media strength. Sometimes the media is very loose, the gelling is very low. Sometimes the media is very hard. It's mostly because of quality adding of agar. The required agar strength we should know before mixing the agar. Different companies supply different kinds of agar, so we have to check before using as a commercial purpose. And change of pH also leads to this improper media strength. And if we over sterilize the media in autoclave, the media charring will happen like it will turn from white to brown color due to this higher temperatures. So we should use optimum temperatures only in autoclave conditions. And the contamination of culture media before and after inoculation. Without inoculation, if we culture contamination is happen in the sterilized bottles, means we have to check the efficiency of autoclave, and we have to check our environmental lab environment and hygiene condition. And to prevent that, we have to use wrappers, seal the bottle cap properly so that we can prevent before inoculation. After inoculation, if we got some contamination, means we have to check our standard operational procedures, laminar airflow calibrations, and other things. And some of clonal variation also we will discuss later on. And genotypic dependence. Like banana, as I mentioned earlier, there are so many groups are there, but Cavendish groups, AAA groups are very responsive for this tissue culture. Other groups like diploid AB group or this Kovvur group, they are very difficult for larger multiplication rates. If you see here in the pictures, you can see higher multiplication rates in the initial cultures of Grand9, Tella Chakrakeli, and Red Banana. But at the same time, see this Kovvur, this Karpura Chakrakeli, the explant is alive, active, but it's not giving any axillary shoots. In case of Kovvur Bonta, it belongs to ABB group. It forms generally in the initial cultures, it forms clumps, but those clumps, if you use the same higher BAP concentrations, it will degenerate. So we have to reduce the BAP concentrations up to 1/10, so that all these clumps will turn into shoots. Then non-availability of ammonium nitrate. By taking this paper as a reference, we have used nitric acid and ammonium hydroxide in the quantity of, we have added 0.921 ml of nitric acid and 0.793 ml of ammonium hydroxide in the culture media instead of ammonium nitrate. So by adding these two concentrations, we got the required quantity of 1.5 grams of ammonium nitrate, and the cultures are very good in growth. And by using this method, we can eliminate the requirement of ammonium nitrate. These two chemicals, ammonium hydroxide and nitric acid, are freely available in the market. You can purchase and you can add these concentrations for later media preparation, so your cultures will grow as normally. And after incubation, if you see, the explants will start, the layers will come out one by one, and the explants will swell. And after a few days, like 1 or 2 weeks, a single shoot may emerge. That is a good and healthy condition of explants. If we are not done virus indexing for mother plant, at least at this stage, we have to collect this sprouted shoot. As we remove this sprouted shoot in the next cycle, we have to do the viral indexing by using this shoot. So we can assure that these are free from any sort of virus contamination. And first few initiation stages, frequently we have to change the culture to free from the phenolic exhalation. Then afterwards, during the shoot multiplication stages, every 25 to 30 days, once in every 25 to 30 days, we have to transfer the cultures. To prevent the high phenolic exhalation and blackening of basal portion, reduce the rhizome size gradually, remove 0.5 centimeters and culture for the next treatment. And sometimes damaging this apical meristematic portion by plus cut or single longitudinal cut will accelerate the axillary buds and prevent the apical dominance, and we will obtain more shoots from the single explant. And during the next transfers, we have to separate these axillary shoots and inoculate into separate fresh culture medium for further shoot multiplication. Like this, we have to do subcultures 25 to 30 days. And here I have given one statement that the subculturing is done until the required amount of plant needed. But the statement is not correct. We may get many number of plants, but if we keep on continuously subculture these plant materials in an unlimited way, the stomaclonal variations may happen. Stomaclonal variations means the regenerated plants are not prototype with its mother plant. So in this, we have to take care that how to restrict our multiplication cycles up to 7 or 8, so that the very minimum number of variations, 2 to 3% variations, we may get, that is permissible only. If we go beyond 10 or 12 subcultures, the somoclonal variations may also increase from 10 to 25%, which is not desirable. And if you produce such type of plants, farmers may complain and they may file a case also. So the labs should be more vigilant on this aspect, and we have to restrict the multiplication cycles up to 7 or 8. We have to check the cultures frequently for bacterial or fungal contamination. If we got any fungal contamination, we have to remove those cultures. If we got bacterial contaminations with slightly, we can incorporate the media, some antibiotics like ceftazidime and cefixium and other things, and we can successfully multiply these cultures. Plant growth regulators also very important. Initial cultures, the high concentration of DAP, 5 grams, 5 milligrams, 6 milligrams per liter, along with 0.2 milligrams of PAA are required. But later on, the concentrations should be reduced to 2 to 3 milligrams per liter DAP for enhanced shoot multiplication. We have also tested TDZ, metatopolin, kinetin, and other cytokinins also. But among all the growth regulator combinations, we found that 4 to 5 milligram per liter BAP along with 0.18 milligram per liter IAA is highly suitable for this grandine and AAA genome group. For Kovvur and other genotypes, 1 to 2 milligram per liter BAP is sufficient. And you can see the multiplication of grandine from single shoot to multiple shoot, and a very vigorous growth of shoots. In case of Purpur Chakrakeli, actually it's a recalcitrant genotype, very difficult to multiply, but we have standardized some protocols by using different antioxidants and growth regulator combinations. We have checked, and you see from each explant, we are getting nearly 10 to 15 axillary buds. So in bone also we have standardized. After well development of these shoots, we have to transfer into suitable rooting media. The rooting media generally we used off MS media, that is the off strength MS media supplemented with a charcoal and adding 0.5 milligram per liter IBA or NAA will give very good roots within 2 to 3 weeks. So sometimes lower BAP concentrations also induce the healthy and fresh roots. So these cultures can be suitable for hardening. Hardening is the final step, but it's a very important step. This step only determines the quality tissue culture plant material. As we know that all the tissue culture process we will do under controlled environmental conditions, under nutrient-rich artificial medium. But once we brought these sophisticated cultures into ex vitro, very harsh climatic conditions. We have to make a shift from heterotrophic nature of the plants to largely fully autotrophic nutrition. So they can tolerate or bear biotic and abiotic stresses like low humidity, high light intensity, water stress, all those things. So before transferring this rooted culture into hardening media, we have to do primary hardening and secondary hardening, two important aspects. These structures like shade net houses, greenhouses, fan and pad systems, all these things we should equip. For primary hardening, this fan and pod system, greenhouses are compulsory. But nowadays, under shade net houses, by using these low-cost tunnels, many commercial people are producing tissue culture plant material. This is the primary hardened tissue culture plants in progress. After primary hardening, we have to transfer into polythene bags or bigger size trays for further development. This is secondary hardening under 50% shade net condition. Finally, we will get a very high quality, disease-free, uniform size and same age plant material, which are highly desirable for farming. From Tissue Culture Laboratory, Kovvur, working under Dr.YSRHU Horticulture University, we have standardized protocols for Kovvur, Karpura Chakrakeli, Kovvur Bonta, Tella Chakrakeli, apart from Grand Nine. These are all our native varieties, so we have standardized thorough protocol. Again, to enhance the shoot multiplication and to overcome the recalcitrant nature of these native genotypes, we have standardized the alternate protocol by using immature male flower birds as an explant source. In case of Karpura Chakrakeli and Tella Chakrakeli, in this, after last hand opening, we have to wait to elongate this stalk up to 25 to 30 centimeters. Then we have to collect these male flower bud, remove all outer layers. The minimum surface sterilization, minimum pretreatment is required, just only washing, and so the very high expensive mercury chloride and sodium hypochlorite, all these things are not required. Collect the immature male hands from 6 to 12 hands. Then culture these hands in suitable media. Within 7 days, you can see the significant change in explant nature. After 15 days, you can observe the swollen bases and tip drying, then cauliflower-like body emergence, and somatic embryo and formation of shoots from cauliflower-like bodies. You can observe 90 days period. So, from within 3 months, you will get a very good number of shoots in case of Karpura Chakrakeli by using this male flower buds. Then we can use our own old multiplication media for multiplication of this male flower bud originated shoots up to 3 to 4 cycles by using 5 mg per liter DAP and 0.18 mg per liter IAA for good growth. To test the clonal fidelity, we have used intersimple sequence repeated markers and found that all the genotypes are identical to each other. There is no clonal fidelity, deviation in clonal fidelity, and all the plants are 2 to type 2 with the mother plant. Apart from these molecular studies, we have also evaluated these tissue culture plants originated from apical meristems, tissue culture plants originated from male flower buds, along with suckers in plant and ratoon crops as an experiment. And we found that tissue culture plants originated from apical meristems and male flower buds yielding 54 to 55 tons per hectare yield compared to suckers. It is giving only 42 to 43 tons per hectare. So the yields are almost 10 to 12 tons, 10 to 12 tons high, and the male flower bud originated plants also performed equally statistically on par with the tissue culture plants. So, and we didn't recognize any monoclonal variations in this Karpura Chakrakeli. Now this year, based on this experience, we have standardized regeneration of Tella Chakrakeli shoots by using immature male flower hands, and you can see the high shoot multiplication of Telachakar originated from male flower buds, and we also standardized Kovvur multiplication by using this immature male flower hands. And recently, ICAR National Research Center for Banana released this NRCB10 as a Kaveri Kalki. This variety also we have standardized and standardized the production system by using male flower buds and producing the planting material as per the demand of farmer. Apart from the commercial multiplication of these genotypes, recently we have restored this native Amurtapani belongs to silk group by using this immature male flower bud technology only. Due to Panama wilt race 1, this variety is not cultivating in our plain areas and almost vanished from the cultivation and existed only in the high altitude areas of Maredmilli. So we have identified this native Amurtapani based on the minimal descriptor characters, and we have collected the male flower bud, and we have cultured these immature male flower hands based on the experience of earlier standardization protocols. Then we have developed the shoots and distributed to tribal farmers. 3,000 plants we have generated, and we have redistributed under AICRP on tropical fruits scheme, TSP subplan program, so that we can restore this endangered Amurtapani cultivar, and also same time we can provide some sort of livelihood for this tribal farmer. This male flower bud technology actually the very highly advantageous, as the collection of explants are very easy, transportation cost is very low, costly surface sterilization and pre-treatment chemicals are not required. 100% culture establishment, as this is free from any sort of microbial contamination. The establishment is very high, and early response, no phenolic exudation, more than 90% regeneration we got in our native banana cultivars, and the resource saving by reducing the production cycle, and reduction of plant material production cost up to 30 to 40%, and efficient multiplication system for most of the recalcitrant genotypes. These are the two micropropagation techniques we have standardized for banana at a horticulture research station Kovvur by using apical meristems and male flower buds. For the enthusiastic graduates, I can tell that in MIDH scheme, that is in Department of Horticulture, this MIDH scheme is there, in which maximum 1 crore, that is 100 lakhs, is issuing as a subsidy for establishing a tissue culture laboratory. If any enthusiastic want to establish a tissue culture laboratory, Department of Horticulture will helpful for you for constructing and establishing a sophisticated tissue culture laboratory by issuing the subsidy. Just I will show the images how a commercial tissue culture laboratory will be available. Before entering, this is the leg washing area. After cleaning our leg and hands, we have to enter into this chamber. In this chamber, we have to dip our legs into Dettol added water. Then dressing room, we have to wear aprons, headgears, facial masks, gloves, and all other things. Then we have to enter this airflow chamber. In this air shower chamber, through air, all the dust will be eliminated. Then we can work in an inoculation room. Here we can inoculate our cultures, and we have to can do subcultures, rooting, and all other things under laminar airflow conditions. We can see the pressure modules, positive pressure modules established in the walls, so that fresh air will come throughout the room. The autoclave room, then growth room. After inoculation, you have to incubate these cultures in the growth room. After sterilization of media, you have to store in media storage room. Then these are the positive pressure modules and generator facility and fire extinguisher facility for emergency cases. And then hardening structures like primary hardening structures and secondary hardening structures. Bottle washing, used bottle washing area, all these things they have established. So This is the total complete protocol from land to lab, again land, for the commercial production of tissue culture plants. I'm very much thankful for university for providing me this opportunity. Thank you.
Sir, thank you, sir. And
Thank you, Harsha.
explained very elaboratively.
Thank you. Thank you.
Sir, if you have any questions, we'll see the questions, sir.
Yes,
Any
yes.
charts are there.
Yes, yes.
No one is given.
No one sent, I think so.
Yes. Students, any questions, please ask. Sir, I think no questions for
Okay,
today's
okay.
lecture. So the
Harsha, I think
that effective manner you
student has
explained
asked,
everything, sir.
Cost of a single plant available to farmer
Raising,
using tissue culture.
Actually, for laboratory production cost is nearly 8 to 9 rupees per plant. They are selling to farmer 12 to 15 rupees based on the credibility of the laboratory and the production cost, depending on the genotype. It actually it helps in obtaining the higher yields. If you use tissue culture plants, the yields will be more compared to 20 to 25% yield increase will be there, so that automatically farmer will get more benefit compared to sucker usage of suckers. And one more problem is, if you use suckers, as I mentioned earlier, so many problems are there. So by using tissue culture plant material, we can reduce the cost of cultivation by eliminating the need of any plant protection chemicals and like that.
Thank you, sir.
Okay, sir. Any more questions?
Why all the explants cannot be converted in productive explants? We will give same pre-treatment and surface sterilization to all the explants, but the problem here is some of the suckers or explants are already infested with bacterial and fungal contaminations. So by using these chemicals, we cannot produce the 100% exenic culture, but we can produce 70 to 75% microbial free cultures. Completely, it's not possible. If we increase the concentrations of this surface sterilization and pre-treatment, then explant mortality will be more due to chemical toxicity. So via media, we can produce 70 to 75%. It's a good number.
Okay, sir. Any questions? Any more?
How can we achieve 100% productivity?
100%. Nagu.
Sir, shall I end the session?
Madam, actually, question అడిగారు మేడం, దాన్ని ఇంకా ఎక్స్ప్లెనేషన్ ఇవ్వలేదు సారు.
Okay.
ఏది హర్ష?
సార్ ఒక క్వశ్చన్ ఉంది సార్ చాట్ లో. సార్ వన్ క్వశ్చన్ ఇస్ దేర్. It is possible to achieve 100% success. That is the question.
How can we achieve 100% productivity?
100% productivity. Yes,
By
sir.
using meristem culture only, we can get 100% productivity.
Okay.
Normal shoe tip culture, it's not possible. We have to exercise the meristematic tips. So under microscopic conditions, so the meristem, the rate of cell division is very high in meristems. So by using meristems as explains, we can get 100% culture establishment.
Okay, sir. Sir, I think all questions are cleared.
Yes.
Okay. Shall we proceed, sir, for the vote of thanks?
Yes.
Sir. So, on behalf of organizing committee, College of Horticulture, Venkataramanagudam, it is my privilege to extend my heartfelt thanks to Dr. Ravindra Kumar, sir. Sir explained micropropagation techniques in banana lecture very elaboratively. Sir, you covered all the aspects in the micropropagation techniques in of banana, like media preparation, explant preparation, media sterilization, stock solutions, that is very important, how to prepare the stock solutions, and hardening techniques. So the problems occurred in the procedures, all are explained, sir, very effectively, and it is very helpful and well understood by our students. Thank you, sir. Thank you very much for accepting our invitation and giving a good lecture on the micropropagation techniques of banana. And I also express my sincere thanks to all the participants, especially to the students, the staff members, and from the different stations and colleges. Thank you. Thank you one and all for everyone. Thank you very much, sir.
Thank you, Harsha.
Thank you, sir.